next speaker, Dr. Reyk Horland. He's going to be giving a virtual lecture. He is not here. But he is the CEO of Tissues, one of the leading companies of microfluidics, organs on a chip, multi-organs on a chip.
Recording in progress.
And it is a German biotech company, and it has a multi-organ chip technology aimed towards conducting preclinical systemic toxicity studies and efficacy studies as well. So this is one of the companies that is actually very advanced in terms of the multi-organ chips. Is it ready yet or no? Yeah, Dr. Reyk, I'm already introducing you here to the audience. I hope you heard what I introduced already.
Certainly not, but go ahead.
Okay, yeah. As I said, Dr. Reyk is the CEO of the leading multi-organ chip technology company, Tissues, and they have made a lot of advances trying to bring the MPS into the multi-organ chips. And it's a German biotech company. And over the course, his academic career in German arthritis research career and the Berlin group of, he was also Professor Roland Landster. He did, I think, work with him. He pursued the development of tissue models that can mimic human biology in vitro. Since joining Tissues in 2010, he has led the effort to establish and characterize a chip-based vascular model for interdisciplinary teams of engineers, computational biologists, and toxicologists, and et cetera, et cetera. Welcome on board. His talk is going to be on commercializing integrated microphysiological systems, current status, and future potential. We are eager to learn from you, Dr. Reyk. Thank you. You can start.
Thank you very much for the great introduction, and yeah, great to be back. Second presentation in this course. I'm also happy that it's kind of a slightly different presentation this time, one which I don't think I have given very often because usually it's more science-based and science-driven. Science will also, of course, be a part of this presentation, but the focus will also be, I mean, what can you commercialize when you develop something in the space of microphysiological systems? I would like to give you a little bit of an idea on how we approach this and what we see as crucial to commercial success in the MPS field. Now, for those of you, always the first slide, who haven't been present at my first talk, just a couple of quick notes about Tissues. As many other players in the MPS field, we are a spin-off from a university. In our case, from the Technical University in Berlin. They're from the Department of Medical Biotechnology. We are currently around 32, 35 people. Still have very strong collaboration with the university, which will also be, which I will also discuss later in the talk, because this is also an integral and essential part of the research strategy, where we do have at this university 30 associated researchers also working for us. We are very happy that we could secure a strong customer base, both in industry and academia, since we went active on the market around 2014, 2015. We have a strong, of course, like many others, have a strong patent portfolio on our technology. And I think we are one of the few companies who are actually in the space certified according to ISO 9001, which means that we have a total quality management system in place, which is of course also something to consider when you want to commercialize your technology, your platform, your services, and maybe also your products. Now, I would like to start with giving you a little bit of an overview of what has happened in the last approximately 20 years in the field. And as you can see, I'm just going to move this forward. There are so many different projects, so many different groups working on this, that actually there are different and unique systems out there, sometimes systems which are competing in the same space, but also very often systems which are more complementary than competing. And to show that to you a little bit further even, I've prepared this slide. First of all, in the context of this presentation, I will use the definition of microphysiological systems like this, which is shown here, that MPS are microfluidic cell culture devices capable of emulating human biology in vitro at the smallest biologically acceptable scale defined by purpose. The important part here is that we only include in this presentation MPS which are of microfluidic cell culture nature. If you see other definitions for microphysiological systems, sometimes also models are included which don't have any microfluidic part at all. But like I said, in this presentation, just to narrow it down to these systems which have kind of a microfluidic component, this will be the definition of MPS. And if you take that definition, you can then basically set and define 4 different groups of microphysiological systems by using 2 parameters. The first one is, of course, is it a single organ system or is it a multi-organ system? And the second one is it plate-based or is it chip-based? So in this case, on top, you have either the single organ plate-based or multi-organ plate-based system, and these are the chip-based system. And this is actually a case of systems which are not competing, the plate versus the chip, at least in my personal opinion, because they will be used in different stages during, for instance, drug development. You have the huge advantage of these plates here that basically you achieve a higher throughput with these plates. You may, of course, be limited with regards to the complexity of the tissues you can mimic in these kind of plates because of the space which you have available. And on the other hand, you have the chip-based systems, either single or multi, which of course have a lower throughput because they are usually more complex in nature. But this higher complexity, of course, also allows you then to use them later in the drug development stage, late in the preclinical stage, when you probably are down to 10 lead candidates and want to understand and get data of whether you move this to the clinic or you abort it in the preclinical stage. And then, of course, just talking that at the end of my presentation a little bit as an outlook, there are projects which want to develop a so-called body on a chip or patient on a chip system, which can as a kind of minimal organismal level, which requires around 11 organ on the chip, to have kind of this minimal organismal level on the chip and use this to even substitute phase 1, phase 2 clinical trials in the mid to long future. Now, having this definition and having all these different systems, you can now also think, I mean, what can I actually commercialize? Which parts of these systems, what can I do? And basically, there are, in our view, 3 components which can be commercialized. So first of all, you of course have the biology. So if you want that, you could think about commercializing the models which you are developing on these chip systems. Then, of course, the second part, clear, the chips themselves, you can sell them as a product. Then if you need devices to run these chips, yes, you can commercialize these devices and sell the devices. Do you have accessories? Are there tier measurement devices, other devices which may be required or are helping with performing the assays? That can be also sold and developed as a product. And of course, then you have the big service part of the whole commercialization strategy, which means that you can either offer services based on your platform. You can also say, we can do rapid prototyping, we can do new chip designs for you. So there are lots of different options in this field of service-based service-based businesses in the MPS field. Now, the first one, organ models. There are so many different organ models which you can build on an MPS system with lots of different approaches. Starting with the cell sources, you can work with cell lines, with primary cells, iPSC derived models, maybe even with tissue slices, and based on that, you can do a lot of different modelings of different organs, and you can think, of course, about then also selling these models to your clients. If you see that you have a best-in-class model for a certain context of use, then go ahead and try to qualify, validate the model, and you can sell it. The second part, of course, the chips, that is, I think, pretty clear. If you have these chips in your hands and if you have a production capability and also the production standards to produce these chips in a robust way, on a high quality level and a reproducible way, then of course you can sell these chips. And I mentioned devices. For instance, in our case, we have these benchtop versions. We have the robot in our hand. I will also talk a little bit about the robot later and why we believe this is going to be an important part in microphysiological systems assay. And you can think about accessories. I mentioned a tier device. We do have one. We have, for instance, also a heat support. So when you want to do long-term imaging, you need to take the chips out of the incubator, and a number of cell culture models are very sensitive to changing temperature, like the lung model. If you want to understand cilia beat frequency, you have to keep the temperature at 37 degrees, otherwise the beating frequency changes. Same goes for cardiomyocytes. So having something which keeps the temperature is quite helpful there. And then I actually included here as accessories in a way, because these are not organ models, but just the sources for these organ models, iPSCs, which we also sell. We have a small bank of donors, and this is important. These donors, they only not give consent for research purposes for these cells, but also for commercial use. And this is very important if you want to sell these to, let's say, a pharma company, that they have the option of doing something on a commercial level with these cells. Otherwise, they are quite limited to internal research, which may not be helpful if you are in the drug development stage. So I just included them here because they are undifferentiated cells, and not we are not talking about the differentiated tissue models which I have in. And then there are services. I mentioned already, you can offer or could potentially offer design of new chips based on specific requests. You could develop fit-for-purpose assays for clients. Yeah. Then also, of course, everything related to, in a way, moving the service business to the product business and saying, what is required that my customer can basically work with the technology in their own laboratories. So you need to do a technology transfer. You need to do a training. So you need to have people training the scientists of the customer in the use of the chips. And then, of course, you can also think about saying fee for service based. If you have assays based on your microphysiological system platform, you can of course also say, you don't need to do that in-house. We can also perform the assays for you. So there are lots of different options in the service field which can be explored when you think about commercializing your platform. Now, I included this here a little bit to give you an understanding about one of our big or biggest part of the contract or the service department which we offer, and this is contract assay development. And this is basically our template where we talk to the customer and say, we can do something like that. So starting with what kind of cells and tissue models do you want to use? What organ combinations in our case do you want to use? What is the runtime of the assay? What can I do in terms of in-process controls? When I sample out of the system, what do you want measured? And then at the end, when you take out the tissues again, what can be measured as endpoint controls? And this as a whole then basically defines the context of use assay which you develop for the client. And this is just, I think I also had it at my previous presentation, kind of an overview of different assays and different context of use cases. Important to note, because this will be important in the next slide, is this kind of category here, which we always define for the different assays, which is a kind of a level of readiness depending on how much evolved the qualification of the assay is. Now, we see this qualification and also the introduction of MPS-based assays into use by industry as a step-by-step process. Usually, it starts, first of all, as a baseline, you need to have qualified equipment. So I said you have lots of options of, also selling devices, accessories. These devices, of course, if you want to sell them, they at least have to have a CE certification. And they should be, it's a little bit small here, they should adhere to in our case, to the European IQ PQ standards for devices. Now, if you have that as a baseline, you basically have lots of people in academia working with this technology, with these MPS systems, and they do lots of wonderful things with this with the technology. They come up with so many different ideas on how and what they can model. The problem always is, of course, and I think everybody who has worked in the cell culture lab and has read a publication and then tried to reproduce what is written in the publication, that very often this fails, and this is what we call the replication crisis. So what you have to do as a supplier of these models to industry, you have to bring then these academic models into your own laboratory and try to replicate it, also qualify these biological models for a certain context of use. And this should be according to the so-called GCCP, Good Cell Culture Practice Standards. When you have that, you can reach out to pharma, to cosmetics, whoever is interested in these assays, and basically qualify the assay itself, which I showed you previously. So you get the assay, and then you qualify together with pharma, ideally together with a customer or a partner. You qualify the assay. Of course, you can also do it internally, but it's always nicer to have somebody from the outside also, selecting with regards to benchmarking the assay, which compounds you use, and so on. Again, this should be done under GCCP standards. And once you have that, of course, and this is the kind of the ultimate goal, you then move into regulatory validation of the assay, and you basically get a validated assay. Now, I already told you a little bit about it, but just to give you more detail, because it also varies a little bit between qualification in different industries. And of course, the main industry is shown here. That would be kind of the pharma, but I also have a small example on how this could work, because also validation is a little bit different in these industries when it comes to, for instance, cosmetics and chemicals. So first of all, first step is convince the end user to use an MPS-based assay. And this is sometimes not easy. So what we always do or try to do is offer a feasibility study at our site, where they can get some insight into the assay. You add trust if it works. You give a value proposition, and then probably you can move to a certain technology transfer of that assay. And if you have that, then there is, of course, Then there is, of course, this second step, which is voluntary product-driven validation by end users. So the end users basically would then use these assays, first of all, for internal portfolio decision-making, but later on also for data which can then be included in INDs. And this is then, of course, the very nice next step to have this as part of INDs, where the data is accepted and basically the assay is used globally for a specific context of use within a certain harmonized guideline. That would be for pharma. And then, of course, you have cosmetics and chemicals. You have to go to these so-called OECD guidelines. And this is more complex and will probably take more time. In our case, what we did, for instance, here is we started working with a cosmetics company on a smaller scale, and then we kind of, moved or stepped it up and worked with Cosmetics Europe, which is basically the association of all cosmetics companies which are active on the European market. This is important. So it's not only European companies, but it's all companies who are active on the European market. So there's also lots of American companies or Japanese companies, for instance, in this group here. So we are now at the level where we basically are working with them and trying together with them to build assays and then push them into OECD guidelines, which usually requires a ring study and a very large ring study before this is accepted as an OECD guideline as an alternative to a specific minimal test. Now, where are we? I mean, I told you all about the different steps now. If you look at the different stages here, so for academia, we do have, and I think it's even more now. Or at least we get now more than 1,000 scientific publications per year on microphysiological systems, which is a lot. As I said, there are so many researchers doing wonderful work with these MPS-based systems. And then, of course, you have the different MPS suppliers. And at least, and I think we did a market survey this year. There are so many new companies coming up, but there are at least 4 standardized commercial platforms available for these qualified equipments. And you do have actually already, and this is data, but I will show the table later. This is data from 2019, where we actually tried in a group to count that. You have at least, probably even more now, 23 qualified context of use assays in the industry, which are used for internal portfolio decision making. And this is kind of new. If I would have given the presentation last year, you would have seen a 0 here, but actually there is, to our knowledge, or was a first data submission on IND level to FDA, which was also successful. So there is one context of use case where basically the data was accepted, which is absolutely wonderful. And I think everybody in the MPS community is really thrilled that this happened, and basically this can pave the way now for lots of more of IND applications or submissions which include MPS-based data. Now, this is the list. This is from a publication from 2020, which basically summarizes the findings of a workshop which was held in October 2019. Basically, pharma was present, cosmetic chemical industry was present, regulatory bodies were present, MPS providers and also academia were present, and basically they all sat together and tried to identify assays where they could also talk about, of course, where basically they could openly say, okay, guys, we can talk about this. We, as a pharma company, we are using this assay in-house. And this is a list, basically, which we came up with during this workshop. As you can see, lots of different groups. I've highlighted, of course, ours, which are mainly, as you can see, the multi-organ chip systems, except for one, where we also have a single organ model, but lots of different assays, lots of different MPS platforms. And this is really, really good because it shows the diversity and it shows the different contexts of use cases which are possible. Now, Coming to a very important part, which is also can be part of any commercialization strategy. When I started my presentation, I said you always have to think about throughput and complexity of these systems. And definitely something which can help with, first of all, the throughput is an automated system, but it can also help you with dealing with the complexity of a certain model, because it can take over a lot of work which usually a scientist would have to do. So especially also if you think about any kind of ring trial, if you can reduce the redundancy and increase repeatability with these automated system, that is a huge step forward. Of course, like I said, higher throughput testing is possible. You generate way more data, and I will also show you why in a minute with these systems. They can help you with physiological maintenance, and of course, they reduce working hours. If you remember my slide with the assay, there are 4 weeks, and now if you think about a sampling regimen or a substance exposure regimen, which tells you, okay, you have to, at least apply the substance once every day over 4 weeks. This means a lot of work during the weekends. So basically, these automates can also, reduce your workload over the weekends. And of course, they perform everything automatically. So that can be really helpful. In our case, this is an automated system which we actually designed ourselves. And why is there so much data? Of course, because the robot can sample at any time point, so you can generate lots of samples. But we also have an integrated microscope, which is basically capable of taking images from all these chips, which are held at the stage here at any point in time during the assay, which, of course, you can imagine generates lots of imaging data, which can be used in quite some smart ways. And I will also show you examples later on. Now, already mentioned, each robot can, in our case, hold 24 chips. You can actually combine with the software up to 4 chips, up to 4 robots to have like a setup for 96 chips, which is in the range of a small animal trial or even a small clinical trial. This is what I already mentioned. And this is then basically the workflow. Yeah? You pick the assay, you have, let's say, you want to do intestine liver, you tell that to the software, which helps you plan it, the robot executes it, and then you basically just do the analysis. And this is the important point, because basically if you write a software which is specific to a certain device, you can also sell that software. So there are actually also possibilities in the computer science, in the software development to build software for these MPS technologies and also sell that and use that in your commercialization. And of course, everybody talks about it. And I mentioned the microscope. You can do a lot of things then also with the amount of data which you generate, basically by using it for machine learning and AI. So we use it, for instance, the microscope to track, for instance, in this case, liver spheroids in the measure fluorescence by fluorescence, pH and O2 concentration in the chip, and measure that over time, flow control, and you can actually also control certain cell behavior in the chip, in this case, how endothelial cells behave. Now that is the robot, and that is for us also the basis to say, looking into the future, what can you do with these systems, specifically also when you have, as I mentioned at the start, when you potentially in the future have some kind of body on a chip, organismoid kind of chip system in place. And then you could think about having IPS banks and picking specific donors from these IPS banks because of their genetic background, their gender, and so on, and then use that to build preclinical trials and generate data because of the selection of the donors, which you have so far only generated in the clinical stage. So basically, you move a lot of data which you generate only in the clinical trial. Now you could potentially move to the preclinical phase. And The important new features here would be that for the first time ever, you would have biological repeats of the patient's body in place. So if you do a clinical trial, you have a patient which is like n equals 1. Now here you can take a patient in a way from the stem cell bank and you put him like on 4 chips in parallel. So you also have a statistic within the patient. That has never been possible before. You can have the same healthy control from that patient in place. Yeah. Normally a patient is sick, and based on that state, you have to evaluate is the drug working or not. Here, you basically have the option of saying, okay, I have the kind of the healthy control of the patient on the chip in place, and I can basically use that as a baseline to understand how good is my drug actually working. You have access to the biological samples along the trial. It is independent from any kind of patient recruitment and hospital usage because it will run on these potentially automated systems. And you are totally flexible when it comes to the trial size. And as I mentioned, you then generate clinical data at a preclinical stage, which can be a huge improvement, and of course is something which you can then offer as part of the services in any commercialization strategy. And you could even think that further. You could think about how can I use MPS in really personalized treatment. So not only thinking about a bank which holds certain donors, but a stem cell bank which holds you as a donor. And then we put you on a chip and understand, for instance, if you get a certain cancer, on what is the best treatment. But already before you may get the cancer sometime in your life later on. So you already have data of you available, and your physician basically just has to look at it and say, okay, I'm giving you this combination of CAR T-cell therapy plus chemotherapeutic agent 1 in order to, have the best treatment for you. So this would be a really personalized approach, and of course opens up a completely different business segment compared to what I've told you previously. And this is just a summary, you get in the end, if you look into the future and open up all these different segments, preclinically informed advanced therapy and drug development options, you can of course replace a number of animal models with it, and this is important also, of course, you reduced the still high attrition rate in pharma, and may also, of course, then if you reduce the attrition rate, you actually may also reduce the time it takes to bring a drug to the market, which of course is like for any patient who's sick now, they don't want to wait for like the average time of 13 years before the drug is on the market. So this may be reduced and may help benefit any patient who is now really keen on getting new drugs. And I think with this, I'm at the end. And again, my last slide, as in my previous presentation, I would invite you all to come to Berlin next year in June and attend the second Microphysiological Systems World Summit. The first one was held in New Orleans this year. We are very happy that it will be in Berlin next year, expecting around 700, maybe even 1,000 people. It's going to be days of a lot of exciting presentations, research, discussion. And would be great to all see you in Berlin next year. Thank you so much for your attention, and I'm happy to take questions. Thank you.
Wonderful, Reyk. And I'm really envious seeing your automated systems. Can I steal a couple of them?
Yes, you will need like a very big car or a freighter or an air transport, but yes, if you have that.
Thank you. We have about 4 to 5 questions time for, and use the microphones, please. Anybody?
A very good talk, and thanks for accepting to speak at our conference in the next couple of weeks in India for the 3R advances. Okay, so I have one question for you. You mentioned something about replacing the humanized mice. My understanding, I'm a veterinarian, so my understanding is humanized mice also have human gene inserts that represent a human in a way. It could be immune cells, it could be T cells, B cells, or anything like that. So, and then the physiology is not disturbed. So you have liver, kidney, brain, everything, and the circulation is going through the gut, everything is there. In a microphysiological system, which is a challenge to create the entire animal, you do have some of it replicated. What is the challenge here, and what is the timeline you expect for replacing a humanized mice, not regular mice? Regular mice I don't accept. Humanized mice is the closest to an MPS. What is the timeline in your mind? It's a challenging question, so thank you.
I would say, like I had it, I think, in one of the first slides, you need to have these body on a chip, organismoid, minimal organismal chip systems with 11 organs in place, and that is not earlier than 2050. But, you're absolutely right. I mean, these humanized mice, we use them because there is at the moment no alternative. They are definitely not perfect because a lot of the other organs in the mice are still, kind of it's a mice organ in that regard. So you only have a certain part which is humanized, and the rest is kind of still working as a mice liver, for instance. That is not perfect. So there is lots of opportunities here, which I see for microphysiological systems even before we are at this stage when you are able to build an human immune competent tissue system on the already existing chips, because that is at least something where you have a number of organs which are all of human origin in combination with the immune system.
So which means that you think that mouse, even though it is humanized, is still a mouse because of other organs that are mouse organs, but your MPS would be actually not a replacement for animal model, but actually a in vitro human model.
That is a very good question. So basically, there are a number of different approaches also when people think about qualification and maybe even validation of these models, because you usually have two data sets. You have lots of data from animal models, and you have data from clinical trials. And the question is always where to compare it to. So do I want to replicate that I have something which is performing like the animal model, or do I want to have something which may be a black box in a way? Yeah, I don't think people in the industry are afraid to use something which they don't fully understand to the down to the smallest biological level, as long as the correlation to the clinical in vivo data is correct. And for us, that is also the kind of the way we see it on how the system should be benchmarked against the clinical data, because we see a lot that animal models are not reflecting what is going to happen in the human situation. But if you have a system which can do that better, then you should benchmark it against the data which you have from the clinics.
Agree. Thank you very much.
Any other questions? Okay. Now, I have something to, because this is about entrepreneurship. You're a successful entrepreneur. Since 2010 to 2022, you've wonderfully automated your labs to multi-organ chips and everything. Can you give us some insights on how you did it? I mean, you must definitely have had some problems on your way of entrepreneurial journey. I mean, we would love to know what were the things that made you successful building this enterprise.
So, I mean, Tissues was founded in 2010, but that was basically just to have, a legal entity for IP reasons. We also started the project in 2010, and it took us, I think, the first 3 to 4 years to first of all develop the platform, but then also get it on a certain quality level where we are confident that we could then also launch the platform both as a service but also as a product. I think you can start with services earlier because, if as long as you have it in-house, everything in-house, you have better control over everything. But as soon as you offer it as a product, it has to have a certain quality level. Otherwise, you get you just get too many complaints and you just burn in a way the product. And that was a long journey, definitely 3 to 4 years with lots of setbacks. But I think that's typical and everybody probably knows it who has worked in the lab. There will be setbacks. It's just a matter of pushing through and keep on going. The second hurdle actually we encountered was that when we first, very early, I think in 2011 or something, or also until mid 2012, when we talked to industry a lot, where, oh wow, this is really cool, really great, but we don't believe that this is going to work because it's science of the future. So come back in 10 years or something. And then what happened, actually, I had it on on the first historical sketch slide, was that the NIH and DARPA, they set up this huge project in US. And then all of a sudden, because this was set up by these big players, there was a complete change of view in the industry also. And everybody was like, okay, if they give so much money, then this must be something where we also should take a look into. And then also, the discussions changed with industry on how and when and if we would use such systems. So this was really, really interesting to see.
One last question from me. I don't know how much, but you probably raised hundreds of $millions over the years. And how did you convince the investors that you need this kind of money for a technology that we don't even know whether it's going to work or not, right? How did you manage to do it? Because I want to learn from you how to raise funding.
Yeah, no, actually, we are probably not the best guys to ask that, because we are, I think, one of the few companies in the field where the founders are still the owners and main shareholders of the company. So we didn't do any large VC rounds, but instead decided to grow the company by the revenue we are generating. So unfortunately, I think there are other people if you ask Mimetas or Emulate, they are very, very successful with raising money. They will probably be able to tell you more about that than I can do.
Thank you. Okay, thank you very much, Reyk. And this comes to an end now, and I'll definitely be in touch with you. Thank you.
Thank you so much. Take care all. Bye-bye.
Yeah, bye.
Thank you, Dr. Mani, for chairing this session. We'll be moving